Hi, and welcome to this short video where we're going to talk about some of the recent results from our group here in the Barrow Centre at Imperial College London.
Metal organic frameworks are a type of crystalline porous material known as MOFs. These are metal ions or atoms coordinated with organic linkers with two or more coordination sites. And because they have two or more coordination sites, the structure extends in all three dimensions. These types of materials can have the tailored pore size, and there are a large range of metals and organic linkers and still more being discovered to use for this type of material.
Chirality is a type of handiness present throughout nature, from right-handed DNA to spiraled snails' genitals. Natural occurring sugars and amino acids are all one-handedness, and this has profound consequences in how organisms will metabolize oppositely handed molecules. And this can influence how a molecule smells, such as the molecule carbon, where the two mirror images smell like caraway seeds and spearmint leaves.
This has more profound consequences for drugs, where the drugs can be metabolized completely differently. And this is due to the fact that chiral molecules will interact differently with, in the presence of other chiral molecules or macromolecules. Separation of these molecules can be compared to separating molecular-sized left and right hands, and for this, a type of material which acts like a glove is needed. So for this, we're using metal organic frameworks, where these can be compared to like a left-handed glove or a right-handed glove.
The size matching of hand to glove is very important, and this has been shown in other publications where they used different sized test molecules and observed large changes in the chiral separation. Metal organic frameworks can contain structural defects which can loosely be defined as any deviations from an ideally ordered crystal structure. A well-studied class of defective MOFs are missing linker defects, while the multi-coordinating ligands are replaced by mono-coordinating ligands.
And this, usually the mono-coordinating ligands have a lower molecular weight, and so this lowers the formula weight and the density of the framework. These types of defects are quite difficult to characterize. So the three techniques we used were to show that there was a porosity increase, where the solvated DMF, where DMF fills the pores of the material, increases for the more defective frameworks because there was more empty space inside the framework.
2. A higher zinc content by mass due to the reduced molecular weight of the monocarboxylic acid ligands.
And there was also a higher full width at half maxima for the powder X-ray diffraction studies with the more defective frameworks. Significantly, our most significant result was that the chiral separation capacity increased with the more defective frameworks. And to test this, we used a gas chromatograph fitted with a chiral column. So here's like the samples we inject into the gas chromatograph, and they're separated with the chiral column, and then we get two peaks coming out.